move on to our third speaker of the session, Mike Stubbington, and he's going to tell us about simultaneous single-cell measurement of open chromatin and gene expression. And so Mike Stubbington obtained his undergraduate degree in natural sciences from the University of Cambridge, and then worked for 5 years as a research scientist in high containment microbiology for the Health Protection Agency. He then returned to Cambridge to get his PhD in molecular immunology at the Barbraham Institute, and then did his postdoc in computational biology at EMBL EBI, and then later at the Wellcome Trust Sanger Institute. Mike led the Human Cell Atlas team at Sanger until April 2018, and he's now with 10x Genomics with a particular interest in applying single-cell technologies to immunology. And so without further ado, Mike, I'll hand it to you.
Thanks, Angela. Can everyone see my slides? Am I sharing the right thing?
Yes, looks perfect.
Fantastic, excellent. So yeah, good afternoon to those of you on Beijing time, and good morning from Cambridge in the UK, where it's still pretty early. It's really nice to be back at an HCA meeting, even if it's only via Zoom, and thanks to the organisers for inviting me. Yeah, so I'm going to talk about another multi-omics approach today, where we want to think about the transcriptome and the epigenome. And of course, thinking about gene expression has been really important in bulk, but also in single cell resolution over the last few years. But of course, ultimately, the genes that are transcribed when you're looking at a gene expression landscape are the result of upstream regulatory mechanisms, transcription factor binding to accessible chromatin and other things like that. And so if you think about it like that, the epigenetic, epigenomic layer is the input into the system, and the transcriptomic layer is the output that is controlled by that input layer. And if we can understand that better, we can think about how gene expression patterns are set up, what cell types are established, and get an even more fine-grained view of the biology in our systems. And you don't have to take my word for it that this is interesting. This was also considered in the HCA white paper when we wrote it a couple of years ago. And here are just a couple of examples of parts of that text talking about measuring RNA and chromatin profiles at the same time. And something I think is quite interesting to note here is that when we wrote this, we were really only able to think about these joint multi-omic methods at low throughput, and the stuff I'm going to talk about today changes that and changes the throughput at which you can measure these multi-omic analytes. And so really until now, if people have wanted to think about a combination of single-cell gene expression and single-cell attack data, single-cell chromatin accessibility data using the ATAC-seq assay, if you wanted to think about that at high throughput and single-cell resolution, you really had to do two separate experiments. So take one sample, take one aliquot and perform a single-cell gene expression assay on it, take another aliquot and perform single-cell ATAC-seq on it, and then computationally integrate those data and infer linkages between the two data sets. And this has worked pretty well. You know, we've seen some really nice work that's done this, but it does make some assumptions that are not always true about being able to see all of your cell types in both data sets and assign them correctly in both data sets. And it doesn't give you that direct linkage, that direct observation, which makes it much easier to interpret the data and see really what's going on. And so we set out to build an assay that does just that and enables you to assay both analytes in the same single cell, so chromatin accessibility and gene expression. And so that gets us to the situation on the right here, where we really can see both of these modalities in every cell that we look at. So I mostly want to give you some data in this presentation, so I won't dwell on the workflow too much, but just to give you a sense of what's going on and how this works. You start with nuclei. So your input into this assay is nuclei, because then you can get access to the chromatin itself, and you can do the transposition of the ATAC-seq assay in bulk. So you transpose your nuclei in bulk. You then partition those nuclei into droplets, where each droplet contains a gel bead. You collect those droplets, and then within the droplets, you can attach the cell barcodes to the transposed chromatin, and also perform the RT step to make cDNA for the gene expression assay. And then you have your barcoded fragments, and you can go through the standard workflow of making libraries. There's one difference here from sort of the typical single analyte assays, where you perform this pre-amplification step just to make sure that your material is well represented in both samples when you split them to make the libraries. So you make one ATAC-seq library and one gene expression library, and then you sequence them as normal. And when you're doing this process, it really relies on these gel beads. And here you can see what's going on the gel beads. They have two different oligonucleotide adapters on them. One that works in the gene expression assay, where it attaches the cell barcode and the UMI to the cDNA product during the reverse transcription step. And then the other one that attaches the same cell barcode to the attack fragments. And of course, because these cell barcodes are the same for both of the analytes, we can use them to uniquely link those two reads, so the RNA reads and the attack reads, back to those original cells that were in each droplet. So that's a brief view of the assay. Let's have a look at the quality of the data, because it's no good if the data quality aren't helpful. So of course, the first thing that you want to get out of a joint multi-omics assay is that you actually want to be able to measure both things in every cell at good quality. And so this plot shows you on the x-axis the attack signal in every cell. So every point here is a cell barcode from the assay, and on the y-axis you have the RNA signal, the RNA-seq signal. And these are both log 10 scales. And you can see there are two populations essentially here. There are the population in the top right, and these are cell barcodes that are associated with strong attack signal and strong RNA sequencing signal. So this is what you'd want to see. These are the cell barcodes that have good multi-omic signal. And then the bottom left are the barcodes that really lack any signal. And these are just the background signals from droplets that contain a gel bead but no cell. And you can also see that we've colored these. So yellow points on this graph are cell barcodes that we assigned as being associated with cells using the algorithms that call that, and then the blue points are barcodes that were assigned as not being associated with cells. So this looks good. The other thing to say is that there's about 2,700 yellow points up here in the top right, and we were aiming to capture 2,500 PBMC nuclei. So again, essentially all of your data are good quality multi-omic data. So that's a good place to start. What about sensitivity? So we compared the sensitivity of our assay, of the joint assay, with what you get if you run it on the same cells from the same sample in the single assay. So if you look at gene expression, you can see, so the blue curve here is the single assay, and the yellow curve is the joint assay. And you can see that for gene expression, either by number of genes per nucleus or number of UMIs per nucleus, you get very, very similar sensitivity. And it's the same story for the attack signal. Again, the sensitivity is very similar. You're really not compromising sensitivity when you're performing this multi-omic approach. Okay, so that's the data quality and how it performs. But what can we actually do with these data when we have the joint analysis across all of our single nuclei? So I want to show you some examples from PBMCs, a nice, well understood system, so we can just illustrate some things that are happening. So we started by profiling 10,000 PBMCs, extracting the nuclei and just running the assay as I talked about before. And if we look at the gene expression data first, so we can perform dimensionality reduction, project the data into UMAP space as is typical, and we can perform clustering. And you can see on the left here the clusters that come out of the Louvain algorithm. And then we can look at those clusters, look at the marker genes, and annotate them. And of course, for PBMCs, this is very well understood. We know the cell populations we're expecting to see. And so we have these populations on the right of this figure, where we have B cells, T cells, myeloid cells, everything you expect in the proportions that you'd expect. We can then look at the attack data, again, project it down into UMAP space. These are, of course, the same cells, remember. We project this down into UMAP space and annotate the cells. Now, it's slightly harder for attack data. And the way we do it is we look for transcription factor motifs using position weight matrices from the JASPAR database. So we look for those motifs that are accessible in each cluster, and then use prior knowledge of what those motifs mean with respect to cell types to annotate those cell types. And this isn't as high resolution as you get in the gene expression data, really because we just don't have that same body of prior knowledge for annotating attack data sets as we do for gene expression data sets. But when we do this annotation, you can see T cells, B cells, NK cells, monocytes. So At this point, we're pretty much in the same position as if we performed two separate experiments on two separate aliquots of PBMCs. But of course, because these are the same cells and we know they share the same cell barcodes, we can combine the data and perform joint analysis. So the first thing to do is to ask, do our annotations match up? Are we annotating the cells with the same identity in both data sets? And we can draw one of these alluvial flow diagrams where on the left are the annotations from gene expression and on the right are the annotations from the attack data, and every line is an individual cell. And you can see that for the vast majority of cells, we do have these concordant annotations between the two modes. So that's a good place to start. And you can see again, this more granularity, this higher resolution annotation from the gene expression. So what can we do now with these data? Well, we can start to refine annotations kind of in both directions across these populations. So if we start by looking at the data in the attack space here, and again, not particularly granular, we can then go back and annotate it. So apply onto the attack space the annotations for the gene expression data. And now it becomes much more granular. And you can see things like this cloud at the bottom, which is split into two, is indeed split by naive and memory B cells. And so this enables you to put granular annotation onto your attack data and think about what the chromatin landscape, the chromatin accessibility means with respect to these granular annotations. So that gives you more power in interpreting your attack data. The other thing you can do is refine expression in the gene expression space. And so I want to think about T cells for a minute. And if you look back at the clustering plot that we had for gene expression, you can see that this cluster in the top right is split into clusters 1 and 2, but these clusters are not particularly differentiated by anything that we have a biological interpretation for, and they're split down the middle here as well. But if you look at the expression of CCR7 in this plot, which is here, you can see that both of these clusters are highly enriched with CCR7. They also express CD3. And so we annotated these as naive T cells. And something that's typical when you do single cell gene expression of PBMCs is that it's usually very hard to separate the naive T cells into CD4 and CD8 T cells. We see this a lot, and other people see this as well. And you can see that actually, if you look at CD4 and CD8 expression, the CD4s are kind of in the bottom of this plot of this cluster, and the CD8s are in the top as a cap on top of this cluster. And that's not how the clustering algorithm, the unsupervised algorithm, wanted to split them. It wanted to split them down the middle. So we haven't been able to split out CD4s and CD8s in the naive population. However, if we go back to attack space and look at the naive T cells, so this is attack space, but annotated with the gene expression labels, you can see that the naive T cells are split here into two very distinct clusters. And if we lay on top of these the gene expression data for CD4 and CD8, we can see that indeed the left cluster here is CD4 naive T cells, and the right cluster is CD8 naive T cells. So using the attack data, we have a clean separation of these two T cell types, which means we can go back and re-annotate the gene expression data to contain these, which enables us to maybe interpret that better. And so going back and forth between the data types, you can improve your annotations and your understanding of what's going on. And then finally, for the last few minutes, I want to show you probably the key thing that everyone wants to do with these data, which is link open chromatin profiles to what that means to gene expression. And a nice example of this in the PBMC dataset is NCAM1, also known as CD56. And this is a gene that's highly expressed in a subtype of NK cells. And so if we do a bit more fine-grained clustering, we can split out the CD56 bright NK cells in our dataset and show that in the gene expression landscape, these cells have high expression of NCAM1. There's also some NCAM1 expression in some other NK cells and some CD8s, which we expect, but there's really no NCAM1 expression in the other cell types that we have here. And so we can look at that, and then we can also look at the chromatin accessibility, the attack signal around the NCAM1 gene. So this is the NCAM1 gene at the top, and this is its promoter, and not surprisingly, the promoter is more accessible in the cell types that have more NCAM1 expression. So that, I think, makes a lot of sense, and you can see it down here at the bottom. But also, we can observe another peak, which is here, which is upstream of the NCAM1 promoter, but really seems to be associated with NCAM1 expression. So you can see that it's more accessible in the cell types that have more NCAM1 expression. We can also look at this peak here, which is only accessible, you only have attack signal in cell types where there's very little NCAM1 expression. And so we can perform some statistical tests on these data and identify these feature linkages where you have attack signal linked to some aspect of gene expression signal, and we can plot them as these arc plots. And here you can see the height of the arc indicates the strength of the linkage, and the color indicates whether it's positive or negative. So this high blue arc indicates that this region, this peak, is strongly associated with a positive correlation with NCAM1 expression. And then this peak here is somewhat less strongly associated, but still statistically so, in a negative fashion with NCAM1 expression. And so you might, if you're trying to identify regulatory regions, infer that there's some positive there's a positive regulation here and a negative association here that's worth further studying and thinking about. So that's all I have to show, and I've just hit 15 minutes exactly, so I think that's not too bad. Yeah, so I just wanted to give you an insight into how you can combine these data to improve your gene your annotations and think about the cell types that are present, but also start to get at these cell state specific feature linkages at single cell resolution. And I think that's really very exciting. So with that, I'll finish and say thank you. This is a huge team effort, as is everything that we do. And thank you for your time as well. And I'll be really happy to take any questions.
Great. I mean, this is one of the newer technologies that 10x has just pushed out, right? So it's pretty interesting data that we have here. Do we have any questions from the audience? We have a question here. How to create the gene activity score to match gene expression from single cell ATAC-seq data? There are many methods, and which one do you recommend?
So if that's about how to collect those feature linkages, which hopefully it is. So yeah, so firstly, I'll point you for all the detail you want at the 10x Genomics support pages where we describe the algorithms in lots of detail. But essentially what we're doing, because we want to get those correlations, and because single cell data, both at attack and gene expression, can be somewhat sparse, as I think everyone knows, to make sure we get really nice correlations and we can calculate those, one thing that we do is we share information between cells within clusters. So we use a weighted sharing approach where for every cell we find its 30 nearest neighbors, and we use a weight so that the very nearest neighbors, that cell borrows more information from them, and then as you go further out from it in Euclidean distance, you borrow less information. But that just starts to fill in some of that sparsity and enables you to calculate the correlation between features more accurately. And there's a whole load of figures on the website that shows how this works. But we find that works pretty well to identify these correlative features.
Cool. I think we have a question from one of the other panelists. How does the gene expression in multi-ohm compare to that from single cell? That was going to be my question too.
Yeah, so I can show you, if I can work out how to go back in these slides. It compares really very similarly. Yeah, so this slide you can see, if you look at the saturation curve, so it's sequencing depth on the x-axis, and then sensitivity either in median genes per nucleus on the y-axis or median UMIs per nucleus on the y-axis. They really track in a very similar way. Of course, this is nuclear gene expression, so it is by definition less sensitive than from a whole cell because there's just less RNA in the nucleus. But nuclei obviously have other advantages, one of them being that you can do attack at all, but also you can use frozen samples more easily. And you can still identify the same cell types. So it behaves the same as just single nuclear experiments without the attack, but differently to when you have cells as input.
I'm curious, so previously I remember when single cell ATAC-seq was first developed, they showed that, and you've shown as well, that you can use the ATAC-seq information to call subtypes. Have you guys done the direct, any sort of statistical comparison between, if you classify using the ATAC versus the RNA, what's the overlap, or are there some cells that get classified differently using the two?
Yeah, so this is probably the best figures I have to show this, where really the annotations are very concordant. So you can see, this is the attack annotation of T cells, and then the T cells that are annotating gene expression, almost all of them remain as T cells. Of course, the gene expression, you can split them into naive and memory, which is easier from gene expression than from attack. And you can see it's harder to separate the NK and the effector CD8s in attack, but we can in gene expression. But again, these alluvial diagrams show that 98% of the annotations match in this case, and we've seen this across multiple experiments. It's not unique to this one. And of course, I guess that relies on you having enough information from prior knowledge for the attack to be able to do that annotation, and it's kind of dependent on your knowledge going in to do it. But if you can do it, and if that prior knowledge is good, then yeah, it tends to make a lot of sense.
So is there any, let's say if you treat the attack features and the RNA features together, if you merge the features in, let's say, one entire matrix, are there new things that you can do, not necessarily as like a paired one-to-one, but like as having additional features for each cell? Have you guys thought about what kind of stuff you can do with that?
So I think that's really interesting. I think they're really very different data types, and when you analyze them singly, you need to perform quite different normalizations on the data to get them to behave computationally and to give you what you want. So I think just concatenating the matrices is not, maybe quite difficult to get a good result from, but I think it's certainly a really interesting approach. Like, how do you not just jointly annotate or jointly analyze by going back and forth or looking for correlations, but take the entire data set and do something with it? Yeah, I think that's really interesting. I hope this community will also have fun things to do with this.
Okay, we got another question from the audience. Have you used this combined data to look at temporal changes like pseudotime type of stuff?
Thanks, Andrew. So we haven't looked at that. I mean, one could imagine that could be really powerful if you have the idea of chromatin accessibility preceding gene expression changes, and you're kind of measuring, you can observe two different temporal aspects that are out of phase with each other. I think that could be really promising, but we haven't done that yet.
Okay. Okay, I think that's all the questions that we got. Actually, I got another question for Dr. Nguyen. I don't know if he's still online and he can answer the question, because we still have some time before the entire session ends. Oh, no, the question was removed. Okay, well,
no, sorry, I just saw the question, so I typed the answer there.
Oh, you typed the answer.
Yeah,
okay,
yeah,
perfect.
Yeah,
yeah.
But I can repeat it if you like.
Yeah, I mean, I think we still have like 5 minutes in the session.
Right.
So I think since all three of the speakers are here, if there are any lingering questions for any of us, I think this is a good time to ask them. So yeah, Juan, do you want to go first?
Yeah, so the question from Ram, and that's actually a really good suggestion that we can combine NicheNet, a new algorithm for testing cell-to-cell interaction that takes into account the downstream changes of the regulation due to ligand and receptor binding into spatial cell-cell interaction prediction. So he names it as spatial NicheNet, which I think is very interesting suggestion. Definitely we will give it a try. Thanks, Joram.
Okay, cool. Any other questions? If not, I guess we can also just, I did get one more. Oh, it's for me. Have I looked at SNP SNV calling at all, link between DNA and RNA? Thank you for that question. Yes, so I'm a little bit struggling with that because I would love to be able to call SNVs using our data, and we did run it through like some existing software to call the SNVs. It looks like we are able to call them for like on the tumor, on the glioblastoma, we're able to call some SNVs. And some of them did indeed match with like the bulk sample that was sequenced. But I think the problem with single cell whole genome sequencing data is like that it's very difficult to know what the ground truth is.
And given the sparseness of coverage when doing whole genome from single cell using any existing method, I'm not sure I've convinced myself how confident I feel in those SMVs. So we're going to continue working on that computationally and see if there's some way we can feel more confident about the calls. But yeah, I mean, we would love to do that, and we can call them, but I just don't know if they're correct. So thank you for your question.
Angela, I have a follow-up question to that. So how
Sure.
about the structural variants? It would be maybe more feasible to call structural variants from the single-cell DNA sequencing.
Yeah, that's a good suggestion. We haven't looked at that yet, but that's a good suggestion. We should definitely try to look at that. Any other questions? If not, then I guess we'll end this concurrent session.